This unit will be on evolution. We're going to work our way through the theory of evolution and natural selection and how this concept came about. And then later in the unit, we'll be closely linking it to genetics and genes in populations, which we've already discussed some. And you'll see how evolution and genetics are very closely linked. I'm sure you've heard of Charles Darwin. Darwin was a well-educated English theologian, and his parents wanted him to study medicine, but he didn't like it. So instead, he became a dedicated naturalist and was asked to travel around South America on the HMS Beagle, which is the ship. And his job was to make observations of the natural world around him. So he made meticulous observations about living species that he encountered. There was a great diversity among living things around the world, and similar species could be found in different places. This was one of the observations that Darwin made. He noticed that there was a wide variety of different species and that some of them looked similar, and those were not necessarily found in exactly the same regions. For example, the mourning dove on the right has very similar features to the Eurasian dove on the left, although they live on different continents. Other observations include similar organisms showed variations within the same area. So, for example, these anteaters both live in South America in the same area, but the lesser anteater on the left maxes out at 18 pounds, while the giant anteater can get up to 100 pounds. So living in the same area, they seem similar, but they show some variation. The remains of ancient organisms were similar to present organisms, but not exactly the same. For example, here's a fossil of an ancient organism that is similar to the modern-day horse, but the number of toes varies. And Darwin found this by collecting a very large set of fossils and then comparing those fossils to organisms that were currently living. So what other ideas influenced Darwin? He wasn't the only scientist that was working on some of these observations at one time. While he was making observations about living organisms, other scientists were observing aspects of the natural landscape and populations. Thomas Malthus, for example, was an English economist, and he proposed that populations outgrow their environments, causing some individuals to die. He was specifically talking about humans, but Darwin related this to other living organisms as well. For example, this mass of frog eggs contains hundreds of offspring. There aren't enough resources of space and food for them to all survive. So some of those individuals are going to die because the population is outgrowing its environment. James Hutton and Charles Lyell were geologists that suggested the Earth's surface was the result of many small, gradual changes over a long period of time. And that theory was called uniformitarianism. So here's an example of this. This is Antelope Canyon in Arizona. Although most scientists in Darwin's day believed the Earth didn't change very much because they weren't witnessing it on a daily basis, those changes were so slow, Hutton and Lyell would have said this canyon was evidence of water eroding the sandstone over long periods of time. So this was one of the first opportunities that scientists took to describe the length of time in which things were happening, that they were things were happening more slowly than they could actually observe in their lifetimes. Jean-Baptiste Lamarck is another scientist that was making observations during Darwin's time. He suggested that traits an individual acquired over its lifetime could be passed down to its offspring. So here's an example of this. According to Lamarck, this heavyweight lifter would have children with above average muscles because he was acquiring larger muscles throughout his lifetime and then passing those traits down. What do we know is wrong with this explanation? So we know that this is not true for this specific weightlifting example. And the reason is because unless the genes in the gamete or the sex cells, sperm and egg, are changed, the traits cannot be passed down. He is not passing down all of the genes and all of the environmental influences on his body. He's just passing down the genes that are going to be transmitted through sex cells. So much of Lamarck's theory was later found to be unsupported. Alfred Russel Wallace is another scientist that came to the same conclusions about evolution as Darwin. He published papers at the same time as Darwin, but never received the notoriety that Darwin did. So lots of scientists working on observations of the natural world at the same time, and Darwin used different aspects of those theories from each scientist to help create his theory of evolution. In summary, Charles Darwin was a naturalist that observed variations in species while on a trip to South America. Influenced by other scientists of the age, he made conclusions about the great diversity he saw in species. Sometimes the theory of evolution and the theory of natural selection are used as interchangeable phrases, and that's not necessarily the case. So let's look closely at the theory of natural selection. Darwin began to use data from other scientists, as we just discussed, and his observations to propose an idea of how life became so diverse. Darwin noticed finches, which is a type of small bird, had beaks specialized for their various sources of food. So this was one of the observations that he made while he was on the HMS Beagle. And from those observations, he theorized that one species of finch had beak variations. He proposed that over time, new finch species had developed different beaks in order to survive on different food sources. So his theory was that one population of finch that existed a certain amount of time ago had a variety of beak shapes. And over time, they had specialized so that there were several different species, each of them with a different beak shape, so that they had changed and adapted to their environment based on their food sources to become then similar but distinct species. So Darwin proposed 3 theories from those observations that changed the trajectory of science to this day. Number 1, individuals compete to survive. Number 2, individuals with the best fitness have a greater chance of survival. This is otherwise known as survival of the fittest or natural selection. And as organisms have adapted over time, they have changed. And this theory is known as the descent with modification. Darwin published his theories 23 years after making them in a book called On the Origin of Species, and he continued to test his theories throughout his lifetime. So we know that Darwin used his observations from his tour of South America, as well as the theories of other scientists. But artificial selection was also being used by humans during Darwin's day, and that is one of the things that led to the natural selection theory. So Darwin's idea of natural selection came from the process of artificial selection, which had been commonly used by farmers. By choosing livestock and crops with the most beneficial traits, they could perpetuate certain varieties. So here's an example. Brassica oleracea is commonly known as wild mustard. From this original plant, right here, farmers have cultivated 6 different vegetable subspecies: Brussels sprouts, cabbage, kale, broccoli, cauliflower, and kohlrabi, by artificially selecting for various traits. So if a plant from this original ancestor had a particularly beneficial yield of flowers, then they might be cultivated throughout time to become more of the broccoli that we know now. Or if they had a particularly beneficial leaf pattern, they might have been cultivated into Brussels sprouts. But this is the ancestor species that then has led to the varieties of subspecies that we have today. So what is fitness? We discussed survival of the fittest. What is fitness? Rather than humans selecting certain traits, however, like in artificial selection, natural selection suggests that organisms that are best suited for their environment perpetuate their genes through reproduction. These organisms are said to have high fitness. So fitness is not being used here in the typical sense that we talk about it today, where a fit organism is therefore bigger or faster or has larger muscles or any of those typical characteristics we use for human fitness. Now that might be the case, but in general, fitness in a natural selection context is simply an organism that is well suited to survive in its own environment and therefore pass its genes down through reproduction. So camels, for example, have high fitness in desert environments because they have wide hooves that prevent them from sinking in the sand and carry fat in their humps so they can go long periods without food. The traits that make organisms well suited or fit for their environments are called adaptations. So just as a little reminder summary here, what adaptations does the snow leopard have? It has camouflage. So if it's living in the snow, it has camouflage here that can help it to blend into a snowy or slightly snowy environment. That helps it to become a better predator because it can sneak up on its prey. Sharp teeth, claws, and forward-facing eyes are all adaptations of predators that eat meat. So those are all possible adaptations that allow this snow leopard to be well suited for its environment and its lifestyle. So in summary, based on the human use of artificial selection, natural selection perpetuates traits that make an organism fit for its environment. These traits are called adaptations. As I mentioned, evolution and genetics fit closely together. So how exactly does genetics fit into evolution? Well, Darwin saw variation in organisms, as we mentioned, but he did not understand what caused that variation. He did not have an understanding of genes and DNA and genetics. Modern geneticists have studied alleles in populations and watched the patterns of variation change over time. So, in very basic terms, that's all we're discussing with evolution is change over time. And these genes are able to be studied now that we know more about DNA, and we can watch the patterns of genes change over time. This group of guinea pigs shows variation in their coat color, as you can see there, caused, of course, by their genes. In genetic terms, a species is a group of organisms that can interbreed. So we're going to talk for a little bit here about species and populations so that we can explain how the variation and allele frequencies in a population or a group change over time. So species is a group of organisms that can interbreed and do that successfully and create offspring. A population, however, is a group of one species in a certain place. So as you can see here, although alewife herrings are a species, they can exist in many locations, but a population might only be those found in that particular lake in this picture. So population is a portion of a full species. What is a gene pool? So scientists can track the variation changes in a population by studying the frequency of an allele within the gene pool. So a gene pool is simply a set of all of the alleles within a population. So you can see here we've got raccoons. There's variation within this population of raccoons. And black fur in this situation is controlled by one gene and is dominant over brown fur. So we have very simple genotypes here, either homozygous dominant, heterozygous, or homozygous recessive. And if we use all of those alleles, we set up all these genotypes for each raccoon in the population, and then we just combine all of those alleles together. That is going to be your gene pool. So out of 10 alleles in this population, we've got 5 raccoons, 2 alleles each, so 10 alleles total. 4 are capital B for black fur, and 6 are little b for brown fur. So the allele frequencies are 40% for capital B or dominant, which is 0.4. Okay, so we've divided now 4 out of 10, 40%. And 60% for recessive, for the recessive allele, 6 divided by 10, 60% or 0.6. So that's how allele frequencies are calculated. If these raccoons lived in an area with lots of dry brown leaves, which variation of raccoons would be the most fit? So if we've got dry brown leaves in the environment, the browner of the 2 raccoons would be more fit for its environment, better adapted for survival. It's the raccoon's phenotype, not its genotype, that makes it well adapted to the environment. So its genotype, as a reminder, is this little b, homozygous recessive, but that doesn't make it well adapted to its environment. It's its actual phenotype, what it looks like, that is going to change how it interacts with its environment. In summary, this discovery of genetics allows scientists to study the frequency of alleles within a population's gene pool and how that changes over time. So we know that genetic variation exists, but what exactly causes genetic variation? Well, genetic variation is simply a measure of the genetic differences within a population, and it causes genetic diversity. A population with high genetic diversity is better able to withstand environmental changes. So genetic variation in a population is a positive thing. It gives the population a better chance of withstanding any changes to the environment. Disease or drought or flood or whatever kind of environmental circumstances might arise. So here's an example. These clams are diverse because they have genetic variation, which may allow some to survive if their sandy environments change. So you can take a look at those and make some predictions about which ones would survive if the sandy environment starts to change. Let's say there's a different composition in the sand. It might be darker, it might be lighter. And there are three main sources of new genetic variation. So in order to create genetic variation, it can be caused by mutations, in which a new phenotype is an accumulation of many mutations, but it can be caused by just one. So a mutation, one very simple mutation, as we talked about in genetics, can be a point mutation where just one DNA base is changed. That could change a phenotype for the better or for the worse in the environment. But often it's an accumulation of many mutations that causes the phenotype to change. So mutation is one source of variation. Gene flow is a second source. This is the passing of genes from a different population or species. So one population receiving a new individual or a new phenotype from a different population. So these would be the same species, but the variation would be different in these two populations. And third, sexual reproduction, in which genes are shuffled during meiosis when chromosomes cross over. So that shuffling can create a new phenotype. What is the Hardy-Weinberg principle? According to the Hardy-Weinberg principle, allele frequencies in a population should remain constant unless a factor causes them to change. In other words, a population that doesn't evolve or is at what we call genetic equilibrium. So this is a population in which the allele frequencies or the genetic diversity does not change over time. And there are certain conditions required to keep that population steady and constant at that genetic equilibrium. So 5 conditions are required for this genetic equilibrium to be maintained. First, there cannot be any mutations because, as we mentioned, mutation causes variation. There can't be any immigration or emigration, so no organisms in or out of the population. No natural selection, so even if the environment changes, the genetic frequency, allele frequency is not going to be changing in that population. There has to be random mating, in other words, no purposeful selection of partners because that might shift the population in one direction or another, causing certain phenotypes to be favored or selected against. And we have to have a large population. So as you might imagine, no real-world population satisfies all of these conditions. This would have to be a population that is existing with no outside influence, no mutations, no change in environment, and completely random mating. So no real-world population is satisfying all of these conditions. This is a hypothetical situation. Like Punnett squares, however, it can be used for predictions and statistical calculations. So we can actually compare what the genetic equilibrium should be if all of these conditions are met, and then if it's not if the genetic frequency is not what we would expect here, then that means that something must have changed. And usually we're discussing natural selection. So if the population does not meet all of these requirements and the allele frequencies are not what we would expect if all those conditions are met, then we're showing that natural selection is occurring in that population. So in summary, a population with a high level of genetic diversity has a lot of variation that can be caused by mutations, gene flow, or sexual reproduction. A population without any of those changes is in genetic equilibrium. Okay, now we'll discuss how natural selection changes genes in a population. So specifically here, we're going to talk about single gene traits. For a single dominant or recessive trait, only two phenotypes exist. This should make sense based on our genetics unit. The phenotypes are either showing a dominant trait or a recessive trait. There's only two options. So in this mouse population, fur color is controlled by only one gene. And notice that there are only two phenotypes, light or dark. If natural selection acts upon the population, one phenotype will increase and the other will decrease. So here we have a predator. If this is our original population, light-colored mice are seen more easily and therefore eaten by predators, so they don't produce as many offspring. The dark-colored mice are more fit for the environment, so you can see that phenotype percentages, at least, are changing from pretty even to much more drastically different. The light phenotypes are being selected against, and the dark phenotypes are being selected for. How does natural selection act on polygenic traits? Remember, polygenic means multiple genes are involved in creating the phenotype. So for polygenic traits, a range of phenotypes can exist. There's not going to only be two, because with multiple genes involved, we can have a range of different phenotypes depending on how many of those alleles are dominant and how many are recessive. Most of the phenotypes are in the intermediate range rather than at the extreme, at either extreme. So this creates what we call a bell curve or a normal distribution, with the largest number of any phenotype being somewhere in the medium range. So in this example here, a certain beetle population wing length can range from 5 millimeters to 9 millimeters. But 7 millimeters in the middle is going to be where we get the highest number of individuals. So most of them are going to have 7 millimeter wingspan, and then there are going to be fewer at those extremes. So when drawn in a graph, a polygenic trait takes the shape of a bell curve, as I just said, otherwise known as a normal distribution. And of course, this is called a bell curve because it looks like a bell. So natural selection can affect the fitness of polygenic phenotypes in three possible ways: directional selection, stabilizing selection, and disruptive selection. And with each type of selection, the graph will shift from this normal distribution. So we'll go through each of those. In directional selection, one extreme phenotype becomes better suited for the environment, and the phenotypes shift in one direction. So here's an example. If beetles with long wings can fly faster, they survive better. They're more fit. Over time, there would be more individuals with long wings. So if we're starting our normal distribution like this, over time, the bell curve can shift so that more individuals are surviving in the population with these longer wingspans. And that's known as directional selection. The population is moving in one direction, and it could be either way, but the graph is shifting in one particular direction based on the adaptations of those organisms. In stabilizing selection, the intermediate phenotype shows the best fitness. So if beetles with short wings were slow and beetles with long wings were unbalanced, medium length wings would be favored. So those would be the most fit. Over time, the phenotypes would shift to have more medium length wings if that was the preferred trait, best fit for the environment. Meaning in stabilizing selection here, we would end up with a greater number in the medium range and we'd end up with a lot fewer in the extremes. So this bell curve is going to shift up. And notice that it's becoming narrower because we're getting fewer of those extreme phenotypes. And the third method of changing genes in a population or shifting genes is disruptive selection. The intermediate phenotype in this situation is the least fit. So shifting species here, if we are looking at oysters that live in rocky coastlines come in a variety of colors from light to dark. Light oysters are camouflaged on white rocks, while dark oysters hide in the shadows. Intermediate colored oysters are preyed upon the most by predators. So here the intermediate phenotype, the middle color, is the least fit. These are both selected for. The extremes are selected for. They're going to survive better. And our graph is going to shift so that it decreases in the intermediate range and both extremes are going to increase. And that's called disruptive selection. So in summary, although single gene traits usually only have two phenotypes, polygenic traits have a range. Natural selection can act to shift the phenotypes in several different ways. Okay, we've now discussed natural selection and its effects on allele frequencies in the population, but how does genetic drift differ from natural selection? So unlike natural selection, in which the population changes because some individuals are better able to survive, genetic drift is a change in the population due to random chance. So this is not a better adaptation, this is just a random change from the environment that is causing the population to change, the allele frequencies to shift. Small populations are the most susceptible to genetic drift because the changes in allele frequency are going to be the most evident in small populations. What is a genetic bottleneck? One type of genetic drift event is called the bottleneck effect. When many individuals in a population are killed, the surviving individuals may have a very different allele frequency than the original population. So this insect population has a change in allele frequencies due to a random event. This is genetic drift. So they have an allele, a phenotype frequency that you can see here, an allele frequency, and then a large portion of that population gets randomly wiped out by being stepped on in this example, and suddenly the allele frequencies are dramatically different and you're going to end up with more of these red individuals and fewer of the black individuals. So the allele frequencies have changed, but not because red individuals are better suited for their environment, it's just a random chance. So here is a nice graphic of the bottleneck effect, and the reason that it's called that. If we've got an original population here, and only a few individuals are going to survive, it's like taking an entire population and putting it through a narrow neck of a bottle. Only some of them are going to survive, and it's possible that some of the variations could die out, or that the allele frequencies may change dramatically. Okay, what is the founder effect? So genetic drift can also occur when a few individuals from an existing population colonize a new habitat. The new population may have a distinctly different allele frequency than the original population. So here we've got our original population. If some of these individuals colonize 3 new habitats, for example, here, we could end up with 3 very different allele frequencies, different from each other, but also different from the original population, depending on which individuals migrate to those different environments. So in summary, genetic drift causes allele frequencies to change due to random chance. The bottleneck effect and the founder effect are 2 types of genetic drift that can cause small populations to change. Okay, so we've addressed how allele frequencies can change or how variation in a population can change over time. That doesn't necessarily mean that new species are being formed. Just means that the population is changing and it doesn't look exactly the same as it did when we originally started studying it. But isolation can lead to the formation of a new species. So Darwin didn't actually tackle the topic of how new species are formed. However, geneticists have seen gene pools change so drastically that groups of organisms can no longer interbreed. So these used to be one species, and now the gene pools have shifted so dramatically in 2 different directions that these 2 groups can no longer interbreed. If that occurs, these would now be considered 2 different species. The formation of a new species is called speciation, and it can occur when populations are isolated from each other. So 2 populations, 2 groups of individuals are separated to the point where they are no longer able to breed with each other, which is what makes them a species to begin with. And there are 4 ways that these populations can be isolated, 4 types of isolation that can cause speciation: geographic isolation, behavioral isolation, mechanical isolation, and temporal isolation. So we'll go through those 4 mechanisms one at a time. Geographic isolation is when 2 populations become separated by a geographic barrier. A river, a road, or an ocean, for example. Over time, these populations become so different that they can no longer interbreed. So that might be because there are different allele frequencies in those 2 populations when they separate, or maybe they adapt to their new regions differently until they become, through natural selection, they become so dramatically different that they can't breed with each other anymore. So here's a graphic for that. If we have a large valley here or a river, and the original population spans that entire region, but then they maybe are pushed upwards because they are looking for cooler weather. This one says climatic changes recede suitable habitat to peaks. So they are maybe climbing, looking for cooler weather and getting out of that valley. And therefore the population gets separated on these 2 different peaks. They can become so different that they become 2 different species. Behavioral isolation is when 2 populations have different mating behaviors, so the organisms have no desire to mate. So they have different behaviors and they stop mating with each other. Common cranes do an elaborate dance to court a partner, and that's one behavioral display that can change if the population shifts so that some individuals are doing that behavior and others are not, and the 2 groups are no longer desiring to mate with each other and therefore not interbreeding. That would be behavioral isolation. Mechanical isolation is when the reproductive organs of the 2 groups become so different that they can no longer mate. This would likely be caused by mutation of some kind that got passed through the population. But as an example, obviously an elephant and a mouse would never be able to interbreed. They've got 2 dramatically different sets of reproductive organs. And then finally, temporal isolation. The two populations do not mate with the same timing. So this would be mating. One group mates during the day, one group mates at night, or they mate in different seasons or even different years. In this example, two species of cicadas are capable of breeding with each other and live in the same area, but they cannot interbreed. They are capable of breeding with each other, but they don't interbreed because one species emerges every 13 years and one emerges every 17 years. And since they only live for 2 years, the timing doesn't overlap, and so they don't get categorized as the same species because they are not breeding with each other. How do prezygotic and postzygotic barriers differ? So the previous isolation mechanisms that we just talked about are all known as prezygotic because they occur before a zygote can be formed. So before, zygotic, zygote, so before the zygote. Postzygotic barriers also exist. If chromosomes don't match up, the hybrid offspring don't form correctly or are sterile. So in a postzygotic barrier, the two individuals actually mate, but they don't produce offspring because the number of chromosomes don't match up from the two individuals. A mule is a good example of this. It's a hybrid offspring of a horse and a donkey, which can mate together, but it's sterile because the horse and the donkey do not have the same number of chromosomes. So in summary, if populations are isolated by geographic barriers, behavior, size, or timing, new species can form. Even if mating does occur, postzygotic barriers can prevent the populations from interbreeding.